So let's start with a brief recap of neuronal events. And hopefully you've had a neuroscience class that went into some depth on these events. I just want to provide a brief overview so that we can focus on some of the events that then are linked to an increased metabolic requirement. So I want to analyze neuronal activity with respect to what events cause a need for increased energy. So you will probably remember that the axon of a neuron at the terminal connects at the synaptic cleft with the dendrites of the following neuron. And typically what happens here is that vesicles from the presynaptic neuron bind to the presynaptic membrane and release neurotransmitters into the synaptic cleft. Neurotransmitters bind to receptors in the postsynaptic membrane, and this causes one of several possible events. It could lead to opening of so-called ionotropic channels, so channels that allow ions to move inside the membrane. They could bind to proteins that cause ions to leave the membrane and hyperpolarize the cell, or they could simply bind to so-called metabotropic proteins that simply cause a secondary cascade of events inside the postsynaptic neuron. But the net effect of this activity, which is what is of interest to us, is that it will lead to so-called postsynaptic potentials. And if they're excitatory, they're called excitatory postsynaptic potentials. If they're inhibitory, they're called inhibitory postsynaptic potentials. Either way, what's happening is inside the postsynaptic neuron, there's a change in the concentration of certain ions. Maybe there's an increase of positive ions, if it's an excitatory postsynaptic potential, or there's either a decrease of positive ions or an increase of negative ions if it's an inhibitory postsynaptic potential. Either way, there's a change in the natural resting concentration of ions, which we will then later have to fix to allow the neuron to restart doing its job. As you might remember, if just about enough positive ions enter the dendrites and spread down to the axon hillock, this in of itself will trigger an action potential. The action potential, which is nothing but another influx of positively charged ions at each break inside the myelin sheath, also known as nodes of Ranvier. This will cause essentially a flow of positively charged ions down the axon hillock. The positive ions will reach the terminal boutons of this neuron right here, which might be represented right here. As positive ions travel down, this will open the calcium channels in the presynaptic membrane. The calcium will come in and will cause vesicles to migrate towards the cellular membrane and binding and releasing of neurotransmitter, and we restart the exact same process that we've already seen. Okay, so for us, what I really want to highlight is that all of this happens by changes in the natural concentration of ions or other neurotransmitters in and around the neuron. And see, the reason I highlight this is because we will then have to restore these concentrations to their natural state. And this is what's going to take energy. Now let's look at this from a membrane potential point of view. So what I'm showing in this graph is on the x-axis time, and on the y-axis, I'm showing the membrane voltage. Now the membrane voltage is really always relative to the outside of the neuron. Now, typically, neurons have a negative resting membrane potential. For example, large pyramidal cells tend to have, on average, a membrane resting potential of just about -70 to -75 millivolts. So maybe a neuron is at its resting potential, and then some excitatory and perhaps inhibitory postsynaptic potentials get into the neuron, happen. And if there's just enough excitatory postsynaptic potential, so if just enough positive ions enter the neuron and reach the axon hillock, the inside of the neuron, as these charges, positive charges come in, becomes more and more positive, or less and less negative, if you want. And if it just reaches a threshold, typically somewhere around -55 millivolts, that will start an action potential. Now, once an action potential is started, two events happen in sequence. First, sodium channels open. So what I'm depicting here is, imagine that this band is the membrane of the cell. So this is the inside or the intracellular compartment, and this is the outside or extracellular compartment. And this is an ionic channel. So it's a channel that allows ions, and specifically sodium ions, to diffuse through it. So when the threshold is reached, sodium, so-called voltage-gated ionic channels, open up, and sodium will rush inside the cell. Now, why does sodium rush inside the cell? Well, there are a couple of reasons. First, there's a chemical gradient. There's much more sodium outside than inside. So just by osmosis, sodium will want to diffuse towards where there is lesser concentration. So sodium, just because of this chemical gradient, will want to diffuse inside the cells. Second, as we said, the inside is relatively more negative with respect to the outside. So sodium, which is a positively charged ion, wants to diffuse, is attracted by the inside of the cell, which is relatively negative compared to the outside. So sodium has two reasons to spontaneously, so effortlessly, diffuse towards the inside of the cell. One is a chemical gradient and one is an electrical gradient. Now, of course, as positive charges rush in, what happens to the inside of the cell? You can see it here. It becomes more and more positive with respect to the outside. Now, when we get close to the peak, the second event happens, which is potassium channels. Potassium channels, also known as the delayed rectifier channels, open up. And again, potassium will want, on the other hand, to diffuse outwards. Why? Same two reasons. First, there's a chemical gradient. There just happens to be greater concentration of potassium inside the cell compared to outside. So just by osmosis, potassium will want to flow outwards. Second, now there's the reverse electrical gradient, because all this sodium came in and made the inside positive. Potassium is a positive charge, and so is now attracted to the outside of the cell, which is relatively more negative. And so potassium will spontaneously flow towards the outside. Now notice how much, and as potassium flows out, what happens to the membrane voltage? Well, the membrane voltage goes back to being a little bit more negative. Right? As potassium leaves, so as positive charges leave the inside of the cell, it goes back to being more negative, the inside with respect to the outside. Now, how much energy did it take to do all this? Zero. All that we had to do was to use something to open the sodium channel. In this particular case, just the fact that they're sensitive to voltage increases. So as soon as the voltage exceeds minus 55 millivolts, these channels just open. And sodium will spontaneously flow inside, because there's both a chemical and an electrical gradient that calls sodium to diffuse towards the inside of the cell. Once that happens, this will open the potassium channels, and potassium will spontaneously flow outwards because it has both a chemical and electrical gradient that pushes it outwards effortlessly. It took no energy whatsoever to do any of this. Everything flowed down gradient, so down its spontaneous, effortless path. What takes energy is the fact that now we can't trigger another action potential because there's too much sodium inside and too much potassium outside of the cell. We need to restore the electrochemical gradient. So we need to push potassium back inside and push sodium outside. And this takes energy. To do this, there's the sodium-potassium pump, or sodium-potassium ATPase, which is this protein that you see right here. And see, the job of the sodium-potassium pump, or ATPase, is to push out at every cycle. It pushes out of the cell 3 molecules of sodium and pushes into the cell 2 molecules of potassium. And now as it does this, 2 things happen. First, we restore the chemical gradient. Now there's again more sodium outside and more potassium inside, so that when the sodium voltage gated channels open, sodium will just want to quickly trickle in. Right? And as soon as the potassium channels open, potassium will want to trickle out because of the chemical gradient. Want to diffuse out. Now, on top of that, notice that we've pushed out 3 molecules of sodium and we've pushed in 2 molecules of potassium. So at every cycle, the inside is getting more negative because it loses 3 positive charges, it gains 2. So there's a 1 point of negative, right? There's an increase of how negative the inside of the cell is compared to the outside, thanks to the sodium-potassium pump. So the sodium-potassium pump is responsible for helping restore the chemical gradient, so that protons will want to diffuse down their chemical gradient, so that ions of potassium and sodium will want to diffuse down their chemical gradient as soon as their channel opens. But it also helps remake the inside more negative with respect to the outside. So the sodium-potassium pump is restoring both the electrical and the chemical gradient, which allows action potentials to happen. Okay, so the action potential in of itself, the flow of ions during the action potential, took no energy. What takes energy is to restore things so that we can trigger the next action potential. Now, every time the sodium-potassium pump does 1 cycle and pushes out 3 molecules of sodium and in 2 molecules of potassium, it uses energy. And energy in the brain is stored in the form of ATP, or adenosine triphosphate. There's no fat storages in the brain the same way that there are in the body. So energy is stored as ATP in the brain. And at every cycle, 1 phosphate is lost from the ATP. And so for every cycle of the sodium-potassium pump, 1 ATP becomes an ADP, or adenosine diphosphate, and 1 phosphate, which gets released. And the energy of that release, it will allows the pump to function. In fact, if you look at the budget of where does the energy go in order for the neural system to work, it turns out, and what you're seeing here is the budget, the energy budget in a rodent model. But it turns out that the lion's share of this is really to restore action potentials and to restore excitatory and inhibitory postsynaptic potentials. In fact, just about 75 to 80% of the energy consumption, which is this big slice right here, which is the energy that is needed to restore gradients after an action potential, and this 34% here, which is the energy that is needed to restore the concentration of ions at the synaptic cleft. Remember, excitatory potentials leak positive ions inside the cell. We want to push those back out. We want to restore the natural gradient. We also want to push out any additional ion that leaked inside or outside of the cell during the excitatory or inhibitory potential. So also at the dendrites, we want to restore the equilibrium so that new excitatory and inhibitory potential can happen. So restoring the inhibitory and excitatory postsynaptic potential down here, and restoring the equilibrium electrochemical after an action potential up here is really the bulk of the consumption inside the brain. The rest is something like maintenance of the resting potential. So the resting potential is dynamic, and so it needs some maintenance. Recycling of glutamate, so you might remember from a couple slides ago that glutamate is inside little vesicles. And those vesicles, once the action potential reaches the terminal bouton, migrate towards the cell membrane, bind to it, and release glutamate. So glutamate was released inside the synaptic cleft. Now we have to take it, we have to repackage it, and push it back in. We have to recycle it, sorry, and then push it back in to those little vesicles. Now, as you can imagine, it's not easy to push something to stock a lot of glutamate, a lot of any molecule inside a small space like a vesicle. So it takes energy to do that, because glutamate will not want to be packaged into one vesicle tightly. So in order to do that, it takes energy. And finally, you might remember that what triggers the vesicles to bind to the membrane potential is the fact that as positive charges come down the axon, that will open voltage-gated calcium channels, which will lead to an influx of calcium, and that will cause the vesicles to bind to the membrane and release glutamate. So we also have to restore the calcium gradient, which means we have to push out calcium. But of course, this too takes energy, because calcium just diffused down its gradient. We now have to push it against gradient, and to do that, we need energy. And these three last things are really this last sort of 20% of energy, somewhere 20 to 25%. So overall, restoring the equilibrium, restoring the state of the cell so that action potential and postsynaptic potentials can happen accounts for just about 75 to 80% of the energy budget, of the neural energy budget. The rest, some 20 to 25% is this sort of other type of housekeeping, maybe maintaining the resting potential, recycling glutamate as needed, and restoring other ionic gradients. Now, it turns out that this was done in the rodent model. The human brain seems to be fairly similar. The only thing that seems to be changing in the human brain is that there seems to be a predominance. So instead of being 47% to restore the action potential and 34% to restore the inhibitory and excitatory postsynaptic potentials, it seems to be reversed pretty much. So this slice here, the restoring of integrative activity at the dendrites, or restoring after postsynaptic potentials, this seems to account for just about 50% or a little more of the energetic budget. And so vice versa, the restoring after the action potentials themselves will actually be less than the number that you see here in humans. So it still amounts to just about some 75 to 80% also in humans, these two slices. Just the magnitude of the two slices appears to be just about reversed in humans. So how is energy produced in the human brain? Well, there mainly are two pathways, two mechanisms to produce ATP. One is known as glycolysis, and this happens in the cytoplasm. And the idea is that if there is glucose, it can be turned into two molecules of ATP. And on top of that, as an additional byproduct of this reaction, two molecules of pyruvate will also be created. Now then, one of two things happen. If there's no oxygen inside the mitochondria, then this will remain what is known as an anaerobic respiration or a non-oxygen-based respiration. So glucose in, 2 molecules of ATP out, and 2 molecules of pyruvate, and that gives rise to 2 molecules of lactate. And that's about it. On the other hand, if there also are 2 molecules of oxygen available, and this happens inside in various parts of the mitochondria, then a chain of reactions is, a cascade of reactions is initiated, which ends up with pyruvate plus 2 molecules of oxygen, leading to 34 additional molecules of ATP. And this is known as the aerobic respiration. Aerobic because it makes use of oxygen. So there are these 2 different pathways, and we're going to discuss some of these items in a little more detail, or rather some of these are relevant to the next discussions we're going to have throughout today's lecture. So really 2 basic mechanisms. There's glycolysis, which turns glucose into 2 ATP and 2 pyruvates. Then if there's no available oxygen, there aren't 2 molecules of oxygen, this turns into the anaerobic respiration, which essentially just turns pyruvate into lactate. If instead there are 2 molecules of oxygen available inside the mitochondria, then inside the mitochondria, you get this chain, which leads to 34 additional molecules of ATP. Now, the thing to appreciate is that glycolysis is done very rapidly. So you can get 2 ATP and essentially 2 lactates very rapidly. Whereas the aerobic respiration takes longer, but of course also give rise to a total of 36 ATP, 34 on average generated through aerobic respiration and 2 generated during glycolysis. So these are the 2 principal mechanisms which give rise to ATP, which then gets used for the sodium-potassium pump and other mechanisms of maintaining the energetic budget of the cells.